Thank you very much first for the kind introduction, and thank you all for attending this webinar about synthetic antibodies, which is my area of research, and it has been for the past almost 10 years now. So before I start dwelling into the topic, I have to tell you a bit more about the main mechanism that's behind the capability of molecular imprinted polymers and synthetic antibodies, which is molecular recognition. Molecular recognition processes are involved in pretty much any aspect of biology, ranging from ligand, receptor interactions to mediate an effect, up to enzyme-substrate recognition to generate products, RNA replication, transcription, and translation into proteins, and last but not least, antigen-antibody interaction, which at the moment, because of COVID-19, is quite popular. And I'm particularly interested in this last one because antibodies represent a very important part of our well-being and our immune system. In particular, what are antibodies? They are proteins, primarily, up to around 200 kilodalton in weight, and they are essential components of our immune system. The first mechanism that basically recognizes something that's non-self and might causes us harm. And because of this, they are tasked with a specific recognition capability. But apart from what they are in this sense, what they represent is also a huge business opportunity, because in terms of the global monoclonal antibodies market, the latest estimate in 2015 was $85 billion, and this market is expected to reach almost $140 billion within the next 4 years. And that is including research type of antibodies, diagnostic type of antibodies, as well as therapeutic monoclonal antibodies. So the potential market for this type of materials is massive. Nevertheless, natural antibodies exhibit limitations because being proteins, they are intrinsically hindered by low stability and very expensive to produce in terms of purity, in terms of storage, and so on. If you look at the literature, if you have worked with them, perhaps you might know that sometimes it's not very easy to actually reproduce data when using antibodies, and there might be some significant batch variation. They don't perform very well in non-aqueous medium, and that's quite an issue when you're thinking about continuous monitoring applications, for example, of organic reactions or something like that. And still in 2020, their production relies on animals. We are talking at least of 1 animal per new type of antibody. And that is just something that's really appalling. Among the alternatives, among the potential possibilities of different type of reagents that might act as an antibody substitute, molecularly imprinted polymers have shown quite a good promise over the years. Now, these are polymeric materials, so they are plastic, but the peculiarity is that they are produced around a target molecule. What for antibodies is the antigen, in molecular imprinting is called the template. So this is your target. And what happens is that you put this target in contact with the forming unit of the final product, which are called monomers. The monomers can interact with the target either by non-covalent interactions, so for example, ionic interactions or dipole type of interactions, or covalently, so forming chemical bonds with the target. Once this interaction has taken place, then this complex is frozen in a network by using a cross-link and performing a polymerization stage. So you transform something that's from a solution into a product that's actually physical, a bulk, for example, a plastic. And at this point, you're left with your target still within your product. The next step is to remove the target, so perform washing steps or peculiar extraction steps so that you can remove the target, and what you're left with is a cavity that's capable in terms of size, shape, and chemical functionality to recognize and associate with the target once again, pretty much achieving the same type of recognition capability that's typical of antibodies. And the advantages of molecular imprinted polymers are several, because they have similar recognition properties to the nature of biomolecules, but they are much, much more stable, being polymers, in harsh conditions such as pH, pressure, and temperature. Theoretically, you can imprint any type of compound, even molecules and proteins that are harmful for animals, for example. So toxins or immunosuppressants, so molecules that are not suitable to be to have antibodies raised against them. And you can use MIPs both in organic and aqueous environments. They are much, much cheaper. They're more cost-effective than biomolecules and can be produced in several formats. And I'll get back to that in a minute. However, all the glisters is not gold, because MIPs are also burdened by some disadvantages. Indeed, sometimes it's not very easy to achieve a full specific binding, because you get a distribution of binding site, especially with classical formats of MIPs, with the at the very early stages of the technology. You cannot get batch-to-batch variability because of this non-uniformity. And one of the main issues is the fact that classically, since the template is in solution together with the ingredients of the production of the MIP, what you get is that you might have an entrapment of the template within the overall structure. And if the washing steps and the extraction steps are not performed very well or not performed to the fullest, then you might eventually end up with template leaking in your during your application, which might, of course, cause contamination and potentially cause, for example, false positives during an analysis, or in any case, artifacts. The fact that you're using template every time you synthesize a batch means that if the template in question is expensive, then of course the all cost of the product might rise significantly. In addition, at the moment, if you are going for this type of products, you need to have somebody who knows what he's doing. So a chemist, a polymer chemist that is familiar and skilled in processing these type of materials. And the fact that classically MIPs have been produced as literally as bulk plastics, it makes it for a poor processability. You'll understand that in a second. So all in all these advantages have historically hindered the application and the translation of molecular imprinting polymers into a more commercial or applicative stage. As I was discussing, MIPs can be produced in different formats. So the historical production has been as bulk monoliths. So you would end up literally with a big piece of plastic that was then ground and sieved into discrete particles, which would then be used for their applications, analytical separations, and so on. Now, this is not very convenient, and although it's very easy, it's still used as a quick and dirty approach to assess the feasibility of a certain type of imprinting process, for example. It has been surpassed by other types of more tailored formats, depending on the application. So, for example, you have films, membranes for separation, for sensing, microparticles for purification applications. And in the past 10 to 15 years, let's say, there's been a huge shift towards the production of molecular imprinted polymers as nanoparticles and nanogels. And you can probably guess why. If we go back at antibodies, as we discussed, they are proteins, they are components of the immune system. But if we look at them from a different perspective, basically they are nanoparticles. So the size of antibodies makes them much more similar to a nanosystem. So it's only natural that people have started to try and get closer to this particular biological element to mimic it. So why MIP nanoparticles? As discussed, nano means very high surface area, which in turn translates into an improved binding kinetics and overall performance, and higher compatibility with, for example, in vitro assays, if you're using enzyme conjugated probes. And the size and dispersibility makes them much, much more similar with the natural counterparts. However, once again, not everything is easy. It's not very straightforward to produce imprinted nanoparticles. There is not a single procedure. It's difficult to optimize the synthetic and the purification conditions. And as previously highlighted, considering that you need to use a new template at each batch, this could rise up the cost of the product and eventually could also leak within the solution, for example, during the application, and you still get a distribution of binding sites, so low affinity versus high affinity MIP nanoparticles. Now, to solve these issues, what we came up with during my PhD was a strategy to prepare these imprinted nanoparticles with a template immobilized on a suitable solid phase. What does that mean? That rather than having the template free in solution together with the monomer units, you would literally fix it, immobilize it on a solid phase such as glass, plastic, and then put this solid phase in contact with the monomers and generate the nanoparticles within this setting. So activate the polymerization process within this setting. This would result in a generation of low affinity and high affinity nanoparticles, but at that point you still have your solid phase with the template, and you can exploit that to separate, to perform an affinity separation process and remove everything that has low affinity and or is not polymerized, is considered an impurity, with suitable conditions, for example, low temperature or a certain pH or ionic strength of your buffer. And once this has been done and the high affinity nanoparticles are still immobilized, are still bound to the target in solid phase, then you give a shock to the system. For example, you increase the temperature abruptly, or you modify the ionic strength suddenly, and you elute the high affinity nanoparticles. So what basically represents your true synthetic antibodies. The advantages of doing all of this is the fact that you are solving the different distribution of affinities within the binding sites, because you are uniforming the affinity thanks to the affinity separation process. The template is immobilized, so it doesn't leak, it cannot contaminate your product, and most importantly, it can be reused. You can perform multiple cycles of synthesis within the same solid phase. The particles can be post-functionalized if you have specific requirements, and I'll get to that in a minute. And thanks to the solid phase synthesis, the whole synthesis can be completely automated. So potentially you would not need a highly skilled polymer chemist to generate this type of materials. So all in all, you are solving the problems that have afflicted MIPs historically. In comparison with antibodies, the costs are much, much lower. We're talking at least 10 times lower, if not more. Preparation time with classically biological antibodies, we are talking about 3 to 6 months, about 4 weeks with the recombinant ones. With synthetic antibodies, you can push at 1 week or even less. The regeneration is very easy and it can be done in mild condition, and physicochemically the stability is quite high. We managed to leave them on a bench for almost, I think, 6 months or a year, and the performance was retained. Plus the fact that they are plastics or they're polymer based means that you can be quite harsh in terms of sterilization processes, so UV, autoclaving, or even gamma rays, for example. In terms of 3 years impact, if you go from this to this, so from biological to synthetic, that means that you can avoid completely at all using animals, and that is something that currently, to the best of my knowledge, is very difficult to achieve when you're talking about antibodies. In terms of how they look, this is the very first image that we ever took of these products. As you can see, they are quite regular, they are spheroidal in shape, and the distribution of size is quite uniform. How do they perform, though, in terms of affinity? Sorry, I think my computer is frozen. Yeah, okay. Sorry about this. Yeah. That's the one. Yeah, that's the one. So they performed quite well. So as you can see, this was the very first test that we've done for these type of nanomaterials, and the target was a small contaminant, melamine, and the binding towards this target was in the performance comparable to their natural counterparts, so nanomolar to subnanomolar, with almost no binding towards the structural analog, which is different only by a few atoms. And similarly, you can have nanoparticles that are produced against, for example, peptides or against antibiotics, even proteins, and you always get, thanks to the affinity separation-based process, affinities within the region of nanomolar to subnanomolar concentrations. As I was mentioning, you can also generate a functional shell within the nanoparticles. So depending on your final application, so you might want to get a more water solubility or water dispersibility. So you might want to add an hydrophilic stabilizer, fluorescent monomers for detection, electroactive monomers, once again for detection, or monomers to increase the possibility of immobilization on surfaces. And how do you do that? The way to do it is basically after the very first step. So after the very first elution in conditions where you remove all the non-polymerized material and all the low affinity material, rather than giving a shock to the system and eluting the high affinity material, you retain them in place and you add a shell-forming monomer. So depending on your needs, you can go, for example, for an electroactive or a fluorescent, and start the polymerization process again. This polymerization process will take place everywhere within your reactor, but mostly as well at the interface on the surface of the nanoparticles, whilst the binding site, conversely, is still protected because it's bound to the target. Then you do another wash, so you remove everything that's not polymerized, everything that has low affinity and so on. And eventually you elute the high affinity, this time core shell imprinted nanoparticles. So something that has a particular functionality on the surface. And we've done that with, for example, vinyl ferrocene, and achieved imprinted nanoparticles that exhibited the electroactive properties. We've done that with the fluorescent monomers, so for example, this eosine acrylate, and the nanoparticles were indeed fluorescent. So that's very important, for example, if you want to think about detection purposes or microscopy applications. And most importantly, as I mentioned, the specificity and the binding, the affinity, were retained in both cases. So you get specific binding much higher than non-specific binding. So the production of the shell did not affect the affinity properties. The other bit that you can play with is, for example, introducing very peculiar monomers, very unusual monomers even. In this case, during this project, the idea was to improve the way aptamers, which are DNA-based affinity materials, are capable of recognizing their target, also in terms of long-term stability. Being DNA-based material, they are susceptible to degradation by enzymes, by DNases, or environmental degradation. What we've done is create the polymerizable variant of an aptamer by exploiting this polymerizable thymine residues that allowed to introduce multiple polymerizable double bonds within the aptamer sequence, and then integrate this aptamer sequence within the imprinted nanoparticles. We've done that by pre-complexing the aptamer with the target, and then polymerizing this pre-complex together with the rest of the monomers, and at that point performing the already mentioned selection step. And in terms of performance, we were quite surprised because actually the aptamers managed, immobilized within the imprinted nanoparticles, gave rise to the best recognition performance in comparison even to the plain MIP nanoparticles and the aptamer alone. In this particular technique, the more negative it goes, the higher the binding, differently from what I've shown you before. And most importantly, we found that the DNA was completely preserved from nuclease digestion. So the fact that the DNA was immobilized covalently within the MIP matrix allowed, or in any case, completely stopped the possibility of degradation by DNases within the MIP. In terms of applications of these type of nanoparticles, you can think of pretty much anything that antibodies are used against. So they've been used in ELISA assays. This was the very first example reported for an ELISA for vancomycin with a limit of detection of 2.5 picomolar. The same assay can be made completely abiotic, and this was done by the group of Professor Piletsky, where they even removed the enzyme from the equation and exploited the redox activity of iron nanoparticles to generate to develop the color for the assay. You can use them as enzyme inhibitors. So in this case, we did produce them against the trypsin, and depending on the orientation of trypsin on the solid phase, you can either achieve inhibition at 19 nanomolar level, or even stabilization of the enzyme, so potentially reducing the autodigestion of trypsin in solution. One of the latest applications that we worked on together with with the Leicester Biotechnology Group was the active targeting and drug delivery. So rather than performing a single imprinting process, we did a double imprinting. So the imprinting on the solid phase was done against an epitope for an EGFR, an epidermal growth factor receptor, which is normally overexpressed in certain types of cancer. And the secondary imprinting, so with the template not immobilized but free in solution, was done using an antitumoral drug, doxorubicin. Once after doing all the usual separation steps, you are left over with a nanoparticle capable of recognizing a target epitope and also loaded with antitumoral drug. So that's actually quite suitable, for example, for anti-cancer therapy. Does it work? It seems so. Indeed, the nanoparticles managed to bind and recognize selectively EGFR when it was overexpressed on certain cell types, such as this MDA468, rather than the SKBR3, which do not overexpress the EGFR receptor, and generated a selective cytotoxic effect on these cells. So certainly there is something that is moving towards potential anti-cancer therapy there. In terms of technology, I hope that I've convinced you that the technology is quite versatile, and it's actually it has the possibility of being conjugated to a number of other technologies. One of our latest reviews showed, for example, that molecular imprinting has been and is in the process of being further investigated together with electrospinning for example, for tissue engineering application or the generation of membranes. And in this context, the usage of nanoparticles is indeed quite interesting, because they could simply be dispersed within the electrospinning solution. So if you're interested about this, we have recently published a progress report that could give you a lot more details about that. And the templates are also quite versatile, because we're not simply limited to small molecules, proteins, or peptides. We are talking even of whole cells, for example. And in these two examples, this is the first one, the authors have managed to actually achieve stem cell differentiation by simply imprinting on siloxane already differentiated cells, and then let the imprinting drive the differentiation of stem cells into specific cell types. Even from, that was confirmed not only morphologically, but also from a genetic point of view. So the genes that were actually activated corresponded to the genes of the differentiated cell lines. And something similar, although a bit more complicated, has been done by Abadi et al. In 2018, where they exploited an hybrid approach of molecular imprinting and nanolithography to actually imprint cardiomyocytes and drive the differentiation of induced pluripotent stem cells into functional cardiomyocytes. So they managed to generate cells that actually were beating and maintained a cardiac rhythm at the end of the project. So that was quite cool. In terms of commercial or potential translation applications, are MIPs delivering? The answer is yes. To what extent, we're not sure. A number of companies, even larger companies, that picked up the MIP technology over the years has been, let's say, significant. Although we still would like to see more MIP-based product over there and being commercialized. The first companies were probably founded around 2003, 2004, and then some of these technology are actually being picked up by big names, such as what was known as Sigma Aldrich, now Merck. And many of these products are still based on either bulk MIPs or micro particles, and are mostly related to separation applications. Whilst one of the latest examples of companies is the MIP Diagnostics, that is focusing on the translation of the nanomips, the MIP nanoparticles, into diagnostic applications. So perhaps something is moving, and certainly there is a certain level of translation, but of course there is a lot that can still be done in this aspect. And I'm all done here, so I would like to acknowledge my funders first, specifically also the NC3Rs for the awarding of a David Sainsbury Fellowship, and all the funders throughout the years for my PhD and postdocs, as well as all the collaborators and supervisors and managers that have helped me throughout these years in achieving these fantastic results. And I thank you all for your attention and for being so patient in listening to this webinar. And if you have any questions, please feel free to ask.